right, welcome back, ladies and gentlemen. So today we are going to pick up talking a little bit about how we figured out what cells even were. So today is going to actually be a little bit of a history lesson, and we're going to talk about these gentlemen here who are considered the major contributors to our understanding of what cells are, how they look, and how they function. Um, you should be able to match the contributions that these gentlemen make to them when we get to the point of taking the test. So make sure that you're capable of doing that. So let's talk through who these guys are. And we'll start with Robert Hooke. He's one of the first major contributors to our understanding of what cells are. This occurred in 1665, and he was an English scientist. And he's most famous for working with cork. And if you know anything about cork, cork is actually a plant. And so he was looking at dead cork cells, as a matter of fact, under the microscope. And the only thing he was really able to see was the actual outer edge of each of those cells, their cell walls. And he looks at them and they're blank on the inside. They've got these sort of square-ish shapes to them. Um, and so he said that they kind of reminded him of the rooms that monks lived in, right? So just these really small square rooms, not a whole lot of furnishings. And what was it that those monks' rooms were called? Cells. And so he used that term cell to describe what he was seeing under the microscope, and the term kind of stuck. So he is the first one to have given these things under the microscope that he was seeing the name cell, and we still use that today. Fast forward just a few years to 1674, and let's talk about this Dutch tradesman. His name is Anton van Leeuwenhoek, and he would trade all sorts of goods all over the world. And something that he just happened to find very interesting to him were lenses. And so he was a tinkerer, and he would play with these lenses and try to make better ones. And how could he make them bigger, smaller, um, higher magnification so he could see more, so on and so forth. And in the process of him playing with those lenses, he put these better lenses into microscopes and was the first person to really study living things under a microscope. And he just kind of used them to study anything and everything that he would pull out of nature and just see what it looked like. Um, and so his biggest contribution was to recognize that these small things that he was seeing under a microscope were actually alive. And so the story surrounding him was that he took a drop of pond water and stuck it under the microscope, and in the creation of these better lenses he was making, he started seeing things. Things were swimming in and out of view and interacting with each other, and he essentially said they looked like small little animals. They knew the term molecule at this point, based on alchemy and some basic chemistry that was going on at the time. And so he sort of shoved the two words together and called them animalcules. Um, we now know there are things like protists and bacteria and things like that that happen to live in the water, zooplankton and all that kind of cool stuff that you can't see with the naked eye. Um, he was the first person to use a microscope to see it. So he termed them animalcules. Term didn't really stick around forever, but it's kind of a fun little story. Then we've got to jump almost 200 years before we see the next big advancement in cellular biology. And that's going to come in 1838 with a German scientist named Matthias Schleiden. And Schleiden, at this point, they understand that living things are made of cells. And so they're just trying to see, well, is it everything that's made of cells or is it just some things? And Schleiden was a botanist, and so every plant that he would come across, he would look at tissue under a microscope. And sure enough, every plant that he observed was made of these same structures called cells. And so he is the first person to propose this sort of overarching generalization that all plants must be made of cells, because at this point he hadn't come across anything different. And we know that today that all plants are in fact made of cells. Fast forward one more year, still in Germany, and Theodor Schwann is basically coming up with the same conclusion except for animals. So every animal that he was observing, again, would take tissue samples, look at them under a microscope, and they would also be made up of cells. And so take the information from Schleiden and Schwann, and we get to the point by 1839 that we can conclude that all living things must be made of cells or cellular products. So, quick tip, how do I keep these two gentlemen apart? Well, Schwann kind of reminds me of swans. Swans are animals, so that's how I remember that Schwann is the one who discovered the animals, and if you can remember him, the other S, Schleiden, the other German, is the one who discovered that all plants were made up of cells. So, quick little study tip there for you. Okay, so next big advancement's going to come in 1855, still in Germany. And we're going to meet this gentleman called Rudolf Virchow. And he's got a really interesting kind of backstory. He's ultimately going to determine that all living cells had to come from pre-existing cells. So they don't just kind of spontaneously arrive. He's going to be one of the first people to put down the idea of spontaneous generation, which did say just that living things just kind of arose out of nothing or out of non-organic material. And Virchow's going to put it kind of a plug in that. Um, so what's his story? Well, he is working at a medical school. And this is sort of the really rudimentary age of early surgeries. And so the doctors are in there trying their best to help patients by performing surgeries on them to fix a variety of ailments, amputations, um, you name it, they're trying it. And what they're finding is that they're losing the vast majority of their patients. Um, but they're not losing them during the surgery itself. Instead, they're losing them post-surgery from infection. And this is also sort of the dawn of germ theory and understanding that there are these small things that exist that can reproduce and grow and metabolize and make us sick. So when you look at early surgery like this, what did they do? Well, they walked into some big room, they cut you open, and that was kind of that. There was no washing your hands, there was no sterilizing of materials. We were using one scalpel on a patient and using it on the next, without much more than maybe rinsing it off. So they didn't understand that bacteria existed, and so they're using these materials from one patient to the next. They didn't understand the idea of the necessity for sterilizing things. And so they're just transferring bacteria all over the place from themselves, from patient to patient, surface to patient. And so most of the patients at this point were dying from post-surgical infection. And so they kept trying to put together, well, what are we doing wrong? What's happening? Why are we losing these patients after the fact? And that's when Virchow sort of comes in and says, there's stuff there, there's living stuff there. And so he, among other scientists working on germ theory at the time, come up with this idea that all cells come from pre-existing cells. And so they're seeing these bacteria that are growing on your hands and on these instruments, and now we're implanting them into our patients, and it's growing there too. And that's what's ultimately killing them. So take the contributions of all five of those gentlemen, and it boils down to one of the major theories in biology, and that's your cell theory. And your cell theory basically boils down to really simple three statements here. All organisms are made of cells. All existing cells are produced by other living cells, and that the cell is the most basic unit of life. That's it. Right? It is probably one of the easiest theories to remember if you can remember those three statements. And so that's going to be a huge component to the foundation of understanding how life exists and how it functions.